Hello everyone, good afternoon all. I welcome you all to today's session. So we'll do PYQ of Principle of Inheritance and Variation. Okay. So we have done till question number 17. So we'll start from question number 18. Okay. So the question is an organism with two identical allele is known as what? Options are A dominant, second is hybrid, option number C is heterozygous, and option number D is homozygous. Okay. So first of all, we need to understand what is an allele. Okay. So this is DNA, right? This is DNA, and we all know that segment of DNA is known as gene. Okay. The segment of DNA is known as gene. Okay. So what is an allele? Then one allele is a specific version of a gene. Okay. Gene means let us take a phenotype of height. So height is the gene. So the variations are one is it can be tall or it can be dwarf. Okay. So for each gene there can be multiple variation and these variations are known as allele. Okay. So first point for allele, you need to understand that allele always present in pair. So for tallness, it can be capital T or capital T small. Okay. So we'll discuss dominant and recessive character for the first is the location of allele. What is the location of allele? Allele are found at the same position on a chromosome, right? Then what are the types? Types are one is dominant and one is recessive. Here, tall is the dominant character and dwarf is the recessive character. Okay. So dominant allele means when the trait. Okay. When the trait. Here, tall is a trait. When the when the dominant allele can express its trait even if only one copy is present. Here you can see here two copies of capital T are present. Okay, but here only one copy of capital T is present, but one capital T can express its character in presence of small t. So capital T here is dominant over small t. Okay, so it is known as dominant allele. Here, capital T is dominant over small t. Second one is recessive allele. That means to express the trait, you have to, that means two copies of allele should be present. Here you can see as two copies of small t are present. So the character dwarfness is expressed. So always allele presenting pair. It can be TT, that means two capital T, one capital T, one small t, or two copies of small t, right? How it is inherited? Okay, it is inherited. One allele is inherited from father, and one is inherited from mother. Okay, so individual inherit one allele from each gene from each parent. Okay, so for example, one is height of plant. Second, second example you can see here is for let's eye color. Okay, eye color you have some of us have brown eyes denoted as capital B. Here capital B is the dominant allele and some has blue eye. Okay, blue eye means small b. Here small b is the recessive allele. Okay, what are the other examples then? Then other examples are blood group we have A, B, O blood group. Okay, here you will see the case of multiple allelism. Okay, that means more than two alleles are present. What are the alleles? One is A, one is B, one is O. So we know we are diploid organism like human, us. Okay, so most genes exist in pairs with one allele inherited from each parent, right? The combination of allele then determine the organism's trait. Small t and capital T determines the tallness. What is the trait here? Trait is the tallness and the phenotypic character is height. Okay, so here you can see the question is an organism with two identical allele. Okay, here two identical allele means the two copies of alleles are same. It can be, in case of height, it can be capital T or small t. Okay, two alleles are same or identical. Okay, so it is the case of what? First one is dominant. Is it the case of dominant? No, because in small t case, it is the case of recessive character. Okay, and is it heterozygous? No, because capital T, small t, when both alleles are different, then it is the case of heterozygous. As two alleles are same or identical, so the answer will be homozygous allele. Okay, what is the answer? You can see here. Answer is, an homozygous individual have identical Mendelian factor or genes for character T or small t that are always pure for particular trait. Okay, if the alleles are identical or same, then the case is homozygous. Okay, then we'll move to question number 19. Question number 19 says, a gene pair hides the effect of one another. The phenomenon is known as what? Okay, a gene pair hides the effect of another. Here, I assume that you have finished the basics of principle of inheritance and variation. So you must know what is dominant. Okay, in basic, dominant means one allele or one allele is dominant over other. That means if one capital T and one small t is present, then the character expressed is in favor of capital T. That is, the plant will be tall, right? So, what is a gene pair? First means gene pair is same as allele. Okay, gene pair means capital T, sorry, small t, capital T, or capital T, small t. These all are gene pair. It is same as allele for a particular gene, right? So, gene pair, one gene from parent, one gene from father, and other gene from mother. Okay, it is same as allele. Okay, then the interaction between these allele determine how the specific trait is expressed. Okay, what is the interaction? Then here, as capital T is dominant over small t, then the character expressed is in favor of capital T, because here capital T is dominant. Okay, so here you can say that answer will be dominance. But what it is it? Here, one allele is, what is, in case of dominant, one allele hides the effect of another, right? In case of capital T and small t, capital T hides the effect of dwarfness or small t, so that it is the case of dominant. That is, capital T is dominant over small t. Because, but in the question, what is given? It is given that the gene pair hides the effect of another. That means two allele, they hide the effect of another allele. Okay. So here in homozygous case, the both alleles of the gene are same. It is case of homozygous case, right? In heterozygous case, two pair of alleles are different. You know, capital T, small t, right? Then what is the dominance relationship? First relationship is known as dominant recessive. That means the dominant allele masks the effect of recessive allele. Okay, in heterozygous pair. So it is the effect of dominance recessive part. One effect, another effect is there, which is known as co-dominance. These are the related topics I'm explaining. Then I'll be explaining the exact effect of gene pair hides another uh, pair. Okay, one gene pair hides the effect of another pair. Okay, so the first concept is dominant recessive concept. That means one dominant allele masks the effect of other recessive allele. Second um, effect is codominance. That means both the allele are equally expressed. Co means they both are equally expressed, right? That means if you will take an example of red flower and smaller, smaller denotes white flower. Okay, it is red, it is white. So RR, if RR allele is present, allele pair, then the flower color will be red. If small r are present, then flower color is white. Okay. So if capital R and small r are present, here the effect of dominance is not seen. Okay. The both red and white, both color are equally expressed. That means the flower color will be pink. Okay. Okay. So it is the effect of codominance. Right. So what is the third effect? Third effect is incomplete dominance. Incomplete dominance. Here the basic units are allele, not gene pair. Okay. If the basic units are allele, then three effects are there. First one is dominant recessive effect. Second one is codominance effect. And third one is incomplete dominance effect. So in incomplete dominance, the resulting trait is a blend of both allele. Okay. This is a blend of both allele. Fine. Then we'll see. Okay. Now one one terminology you need to remember, that if one gene pair hides the effect of other gene pair, it is not dominance, because here not one allele is hiding the effect of other, but one gene pair is hiding the effect of other. So here the effect is epistasis. Okay, effect is epistasis. Epistasis means one gene pair who is dominating, it is known as epistatic gene. Okay, it is a gene pair. Then other gene which is like the expression is suppressed, that gene is known as hypostatic gene. Okay, so in epistasis, one gene which is known as the epistatic gene, it interferes with hypostatic gene and suppress the expression of hypostatic gene. Okay, so two concepts are there. One is recessive epistasis. Okay, and one is dominant epistasis. One is recessive and one is dominant. In recessive epistasis, the recessive allele of one gene masks the effect of another. Okay, we'll go through the examples later. Then in dominant epistasis, the dominant allele of one gene masks the effect of other. Okay, so what are the examples then? The examples are, first one is, there is a coat color of any dog breed. So it is a case of what? It is a case of recessive epistasis. What is the example? That is coat color of a dog breed. Okay, one gene is there. Let's suppose it is gene 1, and another gene is there which is gene 2. Gene 1 is, it has two allele. Okay, two allele means the coat color can be black or the coat color can be brown. Okay, these are the pigments. Then gene 2, which determines if the pigment is there or not. Okay, gene 2 denoted as E, suppose. Then if E is there, then pigment is there. If small e is there, then there is no pigmentation. Okay, so small e results in yellow coats regardless of the B gene. Okay, so here gene E is epistatic gene to gene B. Okay, so if it is the hypostatic gene, then gene E is the epistatic gene. Means if E is there, then there is no pigmentation. That means the coat color is not black. I mean, it is neither black nor brown. It is yellow coat. Okay, so E result in yellow coats, which is regardless gene B. Okay, because here is no pigmentations are there. Okay, so gene E is epistatic to gene B, right? So now you will ask then how it is different from dominance. I already told you that in dominant it occurs between two allele. Okay, in dominance it occurs between two allele, but in epistasis it is involved, like it involves the interaction between two genes. What are the genes? Here the genes are gene B and gene E. It involves the interaction between two genes. Okay, If gene E, e is there, then there is no effect of pigmentation. The pigment is not neither black nor brown, but it is yellow in color. Okay, but it dominant between two alleles. One allele is dominant over the other, right? So the answer is, you can see the answer is A, which is epistasis. Epistasis is the phenomenon of masking or suppressing one phenotypic impression of a gene pair by a non-allelic gene pair. Okay, what is the phenotypic character here? It is the what are the pigments present? Then another phenotypic character is pigments are present or not. Okay, so now we'll move to question number 20. An allele is dominant if it is expressed in option A, both homozygous and heterozygous state, option B, second generation, option C, heterogeneous combination, option D is homozygous combination. Okay, you already know the answer. We already discussed it. So, what is a dominant allele? Let us take a phenotypic character of height again. Okay, so what are the allele? It is capital T for tallness, small t for dwarf. Okay, so capital T, which is the dominant allele, we already know. Okay, so capital T, what are the genotypes? Okay, first is, first write phenotypes, then we will write the genotypes. Phenotypes are plant can be tall or the plant is dwarf. Okay, the plant is tall or the plant is dwarf. What are the genotypes? In tall plant, the genotypes can be capital T, or the genotypes can be capital T, small t. But in dwarf, the genotype is always small t. Okay, so here you can see capital T is the dominant allele, and in dominance, we already know that if only one allele is present, then the character is expressed. Okay, here T, capital T, suppress the expression of small t. That means the character will be tall. So the question says an allele is dominant if it is expressed in, here here you can see T is the dominant allele. It is expressed in homozygous condition, right? It is a case of homozygous. That means two alleles are identical. Okay, or it is expressed in heterozygous as the both alleles are different, right? So the first option is in both homozygous and heterozygous state, which is correct. Okay, what is the second option? Second option is in second generation. Here you can see, if I will cross, this is the first generation. Capital T, small t, small t. Okay, the alleles are capital T here, small t here. So if we will cross, then the product is capital T, small t. Then in second generation, we know we always do self cross. Okay, so here what are the alleles? Capital T, small t, capital T, small. So here capital T, capital T, capital T, small t, capital T, small t again, and small t, small t. Okay, I'm just showing you how the second generation crossing you will do. Okay, but here there is no effect of dominant in second generation, right? So in heterozygous combination, again option number C, and homozygous combination. In heterozygous combination, also the effect of dominant is seen, and in homozygous condition also the effect of dominance is seen. So the answer will be both homozygous and heterozygous state. Okay, here you can see the answer is A, which is dominant allele always express itself in homozygous as well as heterozygous condition. It is denoted by capital letter. Here it is capital T. Okay, so now we'll see next question. A child of O blood group has B group father, then the genotype of father will be what? Okay, so here you can see blood group. Blood group, you know, we have ABO blood group. So we have ABO blood group, right? So what are the alleles? It can be, it is A, B, or O. Okay, so the basics are, if a person has blood group A, that means the allele can be AA or A, I. If a person has blood group B, then the possible alleles can be, possible allele pair can be BB or BI. Okay. If the person has blood group AB, then the person, I mean the allele pair can be A, B. Okay. If the person has blood group O, then the allele pair is I, or you can see IO. Okay. Now we'll do the cross. The child has O blood group. Okay. The child has O blood group. And we know the allele always occurs in pair. One gene, I mean one allele is derived from father and other allele is derived from mother. So here the child has blood group O. That means the genotype is IO. Okay. Here it is written that the blood group of father is B. So it is father. Here I have written mother. Okay. So father blood group is B. That means it can be BB or BIO, right? So if both the pairs are BB, then the child never has I as an allele. Okay. Mother, there is no information about mother. Okay. For father, if there is homozygous condition, then the child will never inherit I gene from the father. You understand that, right? So if the father has BIO allele pair, then there is 50% chances that the child will inherit IO gene. Because if the father has BIO gene, then the allele when segregate, they will form one B, one IO. Okay. So there is 50% of chances that the child will inherit IO gene and the 50% of chances that the child will inherit B gene. But the child has O blood group, that means both alleles are I. So the possible allele pair of B blood group father will be B and IO, which is a heterozygous condition. Okay, BIO means you can write that IBIO. Here AA means IA. Here you can write IAIO. Okay, like that. That means A, B, they are dominant over O. Okay, so if the child has O blood group, that means it has homozygous pair of recessive allele. That means IO. Then the father must be heterozygous, because if the father is homozygous, then the child will never inherit what? The child will never inherit IO gene. Okay, so the options are IO, first option, which leads to blood group O. Okay, but the father blood group genotype is, sorry, the father's blood group phenotype is B blood group. Okay, so the genotype will never be IO. Second is IB genotype can lead to phenotype of B blood group, but for B blood group, as the child is O, as the child has O blood group, so if the father is homozygous, then the child will never inherit IO gene from the father. But we know that from the two allele pair, one pair the child will inherit from its father, and other pair, other allele, he will inherit from his mother. Okay, so option number C is IAIB, which leads to blood group AB, but the father blood group is B. So last option we have IBIO, which is a heterozygous condition for B blood group. Okay, so the 50% chances that the child will inherit IO allele from his father to have a O blood group for the child. Okay, so next, so answer, what is the answer? Your answer is option number D, IBIO. Here you can see the genotype of child would be IO, which is recessive. Hence, the genotype of the father can be IBIO. Then the next question we have, select the correct matches. Okay, so first one is phenylketonuria, which is a autosomal dominant gene. Sickle cell anemia, which is an autosomal recessive trait on chromosome number 11. Then thalassemia, which is X-linked. Then hemophilia, which is Y-linked. Okay. For this, we need to understand that there are some disorders in inheritance and its variation. What are the disorders? Disorders are two types. First one is Mendelian disorder and second one is chromosomal disorder. You will be facing many questions on disorder only. So you need to understand what are the types of disorders and what are the components. Okay. First is chromosomal disorder. What is chromosomal disorder means? There is uh, like in crossing process in meiosis, it is the defect in chromosomes arrangement. Okay, that means in chromosomal disorder, it is due to absence. It is due to absence or excess or abnormal arrangement. Okay, that means when in meiosis we know that there is a step called segregation. Segregation in meiosis 1, it is segregation of chromosomes, and in meiosis 2, it is segregation of sister chromatids. Okay, so when there is when there is a defect during cell division cycle, which results in gaining or losing of chromosomes. Gaining of chromosomes leads to excess chromosome and losing of chromosome leads to absence of chromosomes, or there can be any abnormal arrangement in chromosomes. And others are Mendelian disorder. Okay, Mendelian disorder means it occurs by alteration or mutations in the gene. Here in chromosomal disorders, the abnormal arrangement of chromosomes are there, but in Mendelian disorder, there is a mutation in the gene. Okay, we will see what are the components. That means some examples of Mendelian disorder and some examples of chromosomal disorder. First disorder you will see in NCERT, which is color blindness. So you know in disorders also there are autosomal dominant, autosomal recessive, sex-linked dominant, and sex-linked recessive. Okay, there are four types of disorders are there. Okay, what is color blindness then? Color blindness is sex-linked recessive. Okay, it is a sex-linked recessive disorder. So here the defect is in either, we know that our eyes have rods and cones. So in color blindness, the defect is in either red or green cones of eye, which results in the person can't discriminate between red color and green color. Okay, so as it is X-linked disorder, the mutation present in X chromosome. The disorder is a X chromosome. Okay, and some important facts which you have to remember, that it occurs in 8% of the male and only 0.44% of female. Okay, so next disorder in Mendelian disorder, the next component or next example is, what is the next example? It is hemophilia, which is hemophilia. Hemophilia, it is again a sex-linked recessive disorder. So here you can see option number D is hemophilia Y-linked. Okay, but it is again a X-linked disorder. Okay, so you can eliminate option number D. Then we will see sickle cell anemia. Okay, sickle cell anemia, you can remember like in sickle cell anemia, it is a autosomal recessive disorder. Okay, here there is a point mutation in, you know that hemoglobin has 4 globin chains, so 2 alpha globin and 2 beta globin, right? So here there is a mutation in there is a mutation in sixth position of globin chain. That means in sixth position, glutamic acid, okay, glutamic acid is replaced by valine. Glutamic acid means GAG. Valine is GUG. So here A is replaced by U. Okay, so the glutamic acid is replaced by valine. Okay, so what happens? The normal RBC shape is biconcave. Okay, so the biconcave disc is changed to elongated sickle-like appearance. Elongated sickle-like appearance. Okay, so option number B has sickle cell anemia, which says it is a autosomal recessive trait. Yes, it is a autosomal recessive disorder and which present in chromosome number 11. Here, chromosome number 11, the data you will not find in NCERT, but you can remember that it is present in chromosome number 11. Okay, so the correct option is sickle cell anemia. Okay, now one is thalassemia. Thalassemia, you can remember that it is also a autosomal recessive disorder, but it is written that it is X-linked, so it is not correct. Okay, then one is phenylketonuria. In phenylketonuria, it is a like inborn error in metabolism. What metabolism? First is it is a autosomal recessive trait again. So what are the autosomal recessive trait? Both, sorry, option number A, phenylketonuria, option number B, sickle cell anemia, and option number C, thalassemia. These three are autosomal recessive disorder. Okay, but in phenylketonuria, one enzyme the person lacks. The disorder is in forming the enzyme. The enzyme, okay, so the enzyme which convert phenylalanine to tyrosine. This enzyme is not present. Okay, so there is accumulation of phenylalanine in brain, which leads to mental retardation. Okay, so here the correct option is sickle cell anemia because in phenylketonuria it is written that autosomal dominant trait, but option number A, B, C they are autosomal recessive. The answer is phenylketonuria is an inborn error of metabolism, is also inherited as autosomal recessive trait. Sickle cell anemia, it is also autosomal recessive that can be transmitted from father to offspring when both partners carried the same gene. Okay. Then thalassemia, it is a blood disorder that causes your body to have less hemoglobin than normal, which is a quantitative effect. Okay. Then hemoglobin enables red blood cell to carry oxygen, as we all know that, and thalassemia can cause anemia because there is less hemoglobin than normal. Okay, so if the person will have less hemoglobin, then it leads to anemia. Anemia, what are the clinical features? You will have fatigue. Okay, and in hemophilia, it is sex-linked recessive disorder which shows its transmission from unaffected carrier female to some male progeny. So next we have match the following. Okay, here you can see now we will have to discuss what are the chromosomal disorders. Okay, there are three chromosomal disorders in your NCERT which you need to understand. Okay, so first chromosomal disorder is, here you can write, first chromosomal disorder is Down syndrome. Down syndrome means there is an additional copy of chromosome number 21. Okay, that means there is an abnormal arrangement. Here you can see abnormal arrangement leads to chromosomal disorder. In Down syndrome, there is additional copy of chromosome number 21 is present. Okay, second is Klinefelter syndrome. Here there is an extra X chromosome is present. That means what is our karyotype? 44 autosomes and 2 sex chromosomes are either it is XX in female or 44 autosomes plus XY in male. But in Klinefelter syndrome, here you can find 44 plus X extra copy of X chromosome. That means the karyotype is 47 or 44 plus XXY. 44 are the autosomes. Okay, so the main feature of Klinefelter syndrome that you will find gynecomastia. That means development of breast in male due to presence of extra X chromosome. Okay, so third is Turner syndrome. In Turner syndrome, there is an absence of X chromosome. That means our normal karyotype is 44 plus XX or 44 plus XY. Here, in female, one X chromosome is not there, which means the karyotype changes to 45. That is 44 plus 1X is there, 1X is not there. So, XO. Okay. So, the female will not develop any secondary sexual character. She will be sterile. Okay. So, the karyotype is 45 in Turner syndrome. Right? So here you can see option number C, karyotype is 45, which can be matched to option number 1 in column 2, which has Turner syndrome of sex determination. Okay. So karyotype 45, here you can see it is present in Turner syndrome. Okay. So now we'll understand how we will determine sex. Okay. In sex determination, you can see first in humans. Humans, we all know that it is male. Here I am writing female. Okay. Human have, like, beside autosomes, if it has X and Y chromosome, sex chromosome, then it is a male. If it has two X chromosome, then it is a female. Right? So in case of grasshoppers, okay, in case of grasshoppers, you can see again male has X, Y, and female has X, X. Okay, grasshoppers, or you can also see in Drosophila. In NCRT, you will find Drosophila. But in birds, male has Z, Z sex chromosome, and female has Z, Y sex chromosome. Okay, now you can see a pattern. What is the pattern then? In humans, heterogametes are male. That means heterozygous condition is seen in male. One is X, one is Y. But in female, they are homozygous, XX. In grasshoppers or Drosophila, you can see again heterozygous are male, XY. But females are homozygous. But in case of birds, you can see females are heterozygous. That means one allele is Z and one allele is W. But the males are homozygous. Okay. So here you can match. One is XXXO. Okay. One is XXXO. Second one is XXXY. XXXY method you can see in female homogametic determination. Females are homozygous. That means XXXY method is female homogametic determination. Karyotype 45 is Turner syndrome. Then ZWZZ is female heterogametic sex determination. Okay, here you only write Drosophila. Okay, your NCERT has Drosophila as an example for female homogametes. Female homogametic. Okay, so grasshopper has XX and XO method. Again, XXXO method you will find in grasshopper. Karyotype 45 you will find in Turner syndrome. Okay. Then XXXY you will find in female homogametic, which is seen in humans and Drosophila. And ZZZW you will see in female heterogametic sex determination, which is present in birds. Okay. So next question we have, the answer is D. Here you can see XXXO. They are seen in crickets, grasshoppers, and some other insects. XXXY you can see in females with homogametic sex determination. Then 45 karyotype you can see in Turner syndrome, and ZZZW you can see in birds. So next question we have, in which genetic condition each cell in the affected person has three sex chromosome XXY? Okay, now only I have told you that XXY, it is a chromosomal disorder. It is a chromosomal disorder in which XXY, that means karyotype is 47. With 44 autosomes, you will find XXY. That means the male has extra X chromosome. Okay. So what is the main characteristic feature I have told you? It is, first is the male will be sterile. Second is it has mastia. That means the male will develop breast. Okay, so the answer will be option C, Klinefelter syndrome. Okay, so we'll move to next question, which means thalassemia and sickle cell anemia are caused due to a problem in globin chain molecule synthesis. Select the correct statement. Here you can see in sickle cell anemia, and one is thalassemia. Here, due to point mutation, the GAG is replaced by GUG. That means the biconcave shape will be a sickle cell. Sickle shaped, the RBC shape will change from biconcave to sickle shape. Okay, so here it is a qualitative effect. That means there is no quantity, there is no reduction in quantity, only the quality. That means the biconcave changes to sickle cell. Okay, but in thalassemia, what happened? In thalassemia, what happens? We know hemoglobin has alpha chain and beta chain. Okay, so there is a mutation in globin chains, so that there is reduction in rate of synthesis. Okay, there is a reduction in rate of synthesis. So it is a quantitative effect. Okay, so here you can see both are due to quantitative defect in globin chain synthesis. No, because sickle cell anemia it is a qualitative defect. Second option is thalassemia is due to less synthesis of globin molecule. Yes, because it is a quantitative effect. It refers to decrease in rate of synthesis of globin chain. So it is correct. Third option is sickle cell anemia is due to quantitative problem in globin molecules. No, I have already said that it is a qualitative effect. Both are due to qualitative effect. No, because thalassemia is a quantitative effect. So the correct option is option B. Okay. So next question is a disease caused by autosomal primary non-disjunction. Okay. Here you can see autosomal primary non-disjunction. Just a minute. It is question number non-disjunction. We know that what is non-disjunction. First is it is a genetic phenomenon in which the chromosome fails to separate. Okay. In cell cycle, we know that the chromosome separation or segregation, there is a step. Okay, in non-disjunction, the phenomenon of it is a phenomenon of chromosome, they fail to separate properly during cell division. Okay, which leads to what? Abnormal number of chromosome, right? So it is a chromosomal disorder. Okay, it can happen in meiosis or mitosis. Meiosis and mitosis, these are the cell division. In meiosis 1, we have chromosome separation, and meiosis 2 and mitosis, they have chromatid separation. Okay, so in non-disjunction, the chromosome it fails to separate. Okay, so in Klinefelter syndrome, there is an excess, there is an extra X chromosome is present. In Turner syndrome, there is an absence of X chromosome. In sickle cell anemia, it is due to mutations. But in Down syndrome, I have told you that there is a extra copy in chromosome number 21. It has extra copy in chromosome number 21. Okay. You can also remember that it is first described by Langdon Down in 1866. Okay. So the answer will be Down syndrome. So the next you can see Down syndrome is caused by non-disjunction of 21st chromosome, that is trisomy. That means a additional copy of a chromosome number 21 is there. Next question, which of the following most appropriately describe hemophilia? Okay, I have told you that hemophilia it is a sex-linked disorder, sex-linked recessive disorder. So first option is recessive disorder. Second option is X-linked recessive genetic disorder. Then third option is it is a chromosomal disorder. No, because in chromosomal disorder we have studied one is Down syndrome, one is Turner syndrome, and one is Klinefelter syndrome. And fourth one is it is a dominant gene disorder. No, because it is a X-linked recessive disorder. Okay. In the classification itself, I have told you right. So option B is correct. Here you can see hemophilia is a X-linked recessive disorder. It has two components. One is hemophilia A and hemophilia B. They both are X-linked recessive disorder. Okay. Next is pick out the correct statement. First option, hemophilia is a X-linked recessive disorder. Now only we have seen that it is a sex-linked recessive. Okay. So it is correct. Second option we have Down syndrome. Down syndrome due to aneuploidy. Okay. Now you will see aneuploidy. Aneuploidy means there is a failure of segregation of chromatid during cell division. Okay. Which results in whether you can gain or lose a chromosome. It is known as aneuploidy. Here we have seen that in Down syndrome, what happens? In Down syndrome, there is a trisomy of chromosome 21. That means an additional copy of chromosome is present. So it is a case of aneuploidy. Third option is phenylketonuria is an autosomal recessive gene. Okay, it is also correct because it is an autosomal recessive gene. Sickle cell anemia, phenylketonuria, and thalassemia, they are autosomal recessive. Fourth option is sickle cell anemia is a X-linked. Okay, so it is incorrect because it is a autosomal recessive disorder. So option 1, 2, and 3 are correct. Answer is D. Okay. So next up we have what is a pleiotropic gene. Okay. You can remember it as pleiotropic gene means one single gene. It will the manifestation of one single gene defect are many. That means a single gene it exhibit multiple phenotypic expression. Okay, you can say a single gene it will exhibit multiple effect. Like in phenylketonuria, you know that there is an enzyme which is absent, which convert phenylalanine phenylalanine to tyrosine. The enzyme is absent, so the phenylalanine the phenylalanine quantity in brain is increased, which leads to mental retardation. Okay, as well as you will see reduction in hair and skin pigmentation. Okay, so one single gene leads to mental retardation also, then hair and skin pigmentation also, reduction in hair and skin pigmentation also. That means the manifestations are many. You can see a gene evolved during Pliocene. No. Option number B is control a trait only in combination with another gene. No. It control multiple traits in an individual. Yes. The answer is option number C, which regulates multiple traits. Next is human female with Turner syndrome. We know that Turner syndrome, there is absence of one X. There is absence of one X chromosome, which leads to karyotype 45. That means 44 plus XO. Okay. So option 1 is correct. It has 45 chromosome with XO. Okay. All other options are incorrect because in option 2, additional copy of X chromosome is there, which is present in Klinefelter syndrome. Okay. And the person will be sterile. Answer is A. Okay. Incorrect statement regards to hemophilia. First is hemophilia. We know that hemophilia it is a X-linked recessive disorder. So first one is correct. Second one is it is a dominant disorder. No, because it is a X-linked recessive disorder. So second one is incorrect. Third option is a single protein involves in clotting of blood, which is affected. Yes, in hemophilia, the mutation leads to like the clotting of blood is affected. Okay, that means a single protein that is a part of cascade of protein which involved in clotting of blood that is affected. Okay, it is X-linked disease. Yes, it is a X-linked. It is a X-linked recessive disorder. Okay, so answer is B. We have to know the incorrect options. Next question is which one is the incorrect statement regarding pedigree analysis. Okay, pedigree analysis you have to remember that first is okay. So first is what is pedigree analysis. Okay, pedigree analysis that it is based on inheritability of certain characteristics in families. Okay, we have family tree present. Okay, so it is an analysis of traits in several generations of a family. It do not confirm that DNA is the carrier in genetic information. Here in option number B, it is written that it confirms that DNA is the carrier of genetic information. But you will be reading in next chapter, that is molecular basis of inheritance. That is the Griffith experiment and all experiments. They confirm the DNA is the genetic material. Okay. So the incorrect statement is option number B. Next question. Again, repeated question. Down syndrome is due to monosomy. Okay, Down syndrome is due to, okay, not Turner syndrome. It is written Down syndrome. Down syndrome means trisomy of 21st chromosome. Okay, in Turner syndrome, it is monosomy. That means 45 plus XO. It is monosomy. Here, Down syndrome means 3 copy of chromosome 21 is present. Okay, next, a normal vision man whose father is a colorblind. Okay, here you can see in normal vision man, father was a colorblind, means XXY. Normal vision man means he will inherit Y gene from his father, so he is XY. He marries a woman whose father was also a colorblind, so XXXY. The female will inherit X chromosome, so XX from father, and as she is a normal woman, that other X is normal. Okay, if we'll cross XY, XX, X, then the results are, the possible phenotype of their children are, one is X, XX, XX, XXY, and XY. Female, female, male, male. Okay, the question says they have first child as their daughter. That means the genotypes can be X, XX, and XX. The question is, what are the chances of their child would be colorblind? The female uh, child, the possible phenotypes are, she may be a carrier because only one gene is affected, or she is normal. So the percentage of colorblind is 0. But if it is a male child, then the percentage will be 50%. Okay, because for possible scenario, there is one male child is affected. So answer is B, 0%. Okay, here you can see last question. Which of the following is wrong statement regarding mutations? Okay, mutations means there can be a point mutation. That means only one base pair is affected. Okay, only one base pair is affected, which is known as point mutation. Okay, so deletion and insertion of base pair can cause strain shift mutation. Okay, if there is a base pairs is deleted or inserted, then there is a friendship mutation. Then second option is, in cancer cell commonly show chromosomal aberration. It is also right. In cancer cells, the most common cause is chromosomal aberration. Third option is, the UV ray and gamma rays are mutagens. Yes, we all know that the UV rays and the radiations are, they can cause mutations in organism, so that they are called mutagens. Fourth option is, change in single base pair of DNA doesn't cause mutations. No, if there is a change in single base pair, it is known as point mutation. Okay, there can be a point mutation, there can be a friendship mutation. Here it is written doesn't cause mutation, which is wrong. So the correct answer is option number D. Right. So here we'll end. Thank you so much.